So, good evening, everyone. This is Kanika Gupta, and I'm your TA for the session. We'll do it accordingly as we've been doing from the last few sessions. I'll first give you a preview of what you guys must have read in the last week, and then we'll discuss if you have any doubts or something. And we'll also discuss the questions of the assignment. So, let's proceed towards our lecture, okay? Yeah. So in the last week, basically in the week 10, we learnt about cloning. How do we do cloning, and what is the scheme? What are the things that are involved in cloning, and what are the different types of cloning we can have depending upon in mammalian cells or mammalian vector or something, or yeast cells or yeast vector. So depending upon that, the different types of cloning, and also we learned about their delivery also, कि how we can grow them inside the cell and all. Okay, so this is what we learned in the last week's lecture. Just a moment. This is what we have learned. So let's proceed towards our lecture. Just a moment, class. Yes. Yes. So in the last week, we learned about molecular cloning, ki how it is done, and what are the different things that are involved, that are required for cloning. So first of all, it is we need a genome, and from that genome we need that particular gene that we want to clone. Then we have to cut the clone, we have to cut the vector also with the same enzyme, so that exactly similar sites get formed. And then we will ligate the gene inside the vector, and then it will uh, transform that vector inside the suitable cell, and then it will be grown on the suitable plates, so that there should be some kind of a selection marker inside the vector, so that a particular type of only those cells can be grown in which our vector has gone, our fully cloned vector has gone, not the other way around. Okay, so this was the general idea. This is the general idea of molecular cloning. Steps in molecular cloning. So the steps involved in molecular cloning are, first of all, we need to identify the gene, the genomic library, either from the cDNA library or from the screening part. Then we have to do the gene amplification, cloning, and screening. So cloning is basically cutting the gene and the vector with the same enzymes and getting them inside the vector through ligation. Then the DNA delivery, the cloned vector should be delivered to the suitable cells, and then the expression of overexpression of that particular gene, so that the desired protein can be made. Okay, so construction of genomic library, how it is formed. So basically, the isolation of genomic DNA is done, then the restriction digestion of genomic DNA is done, then the purification of DNA fragments is done, and then the ligation of fragments into the suitable bacteria. Here they have used yeast vector. So in the yeast vector, they have done the ligation part. Just and then the transformation into the yeast cell only, and that is how you form your library. Here they have done the random cutting of the genomic DNA, so that you can get different fragments or different genes from the genomic DNA, and that makes the whole library. From there you can choose your particular gene and then can clone that. Okay. uh, So step one is that we are not going into the detail of the isolation of genomic DNA. I hope you have heard that. Just an overview that first of all, cells are lysed. Then the enzymatic digestion of the digested lysed cells is done, so that this DNA can be cut it down into the smaller fragments. Then the precipitation is done using the absolute alcohol. Then the purification is done by the chloroform phenol method. So chloroform phenol method is done just for the better purification for so that every protein content or any kind of other materials can be removed from the sample. The precipitation with the absolute alcohol and solubilization in water is done. So while you are precipitating in it in a absolute alcohol, then the alcohol must be completely removed after precipitation, otherwise it will hinder in your reaction. It will cause problem for the further amplification and all steps. Okay. Then after the generation of suitable size fragments, the fragments should be generated using the restriction digestion enzymes. In addition to genomic DNA can be fragmented using mechanical shearing also. By mechanical ways also the shearing can be done. But in case of generally it is done using the restriction digestion, so that the random sizes of DNA fragments can be formed, a suitable size of DNA fragments can be formed. Okay. So this is what is done in the generation of suitable size fragment. Then the cloning of those fragments should be done in a suitable vector depending upon your requirements, that in which cells you need that gene further for purification purpose or whatever purpose you are cloning it for. So according to that a suitable vector is again digested using the same enzyme through which you have digested the genomic library genome to make the genomic library, so that the similar kind of ends are formed on both the gene and the vector, and the cloning can be easier. Okay. So the suitable vector to prepare the genomic library can be selected based on the size of the fragment of genomic DNA and the carrying capacity of the vector. Size of the average fragment can be calculated, and accordingly suitable vector can be chosen. In the case of fragment generated by restriction enzyme, vector can be digested with the same enzyme and put for the ligation to get the clone, as I've told you. In case of mechanical shearing mediated fragment generation, putting these fragments need additional effort. In one of the approach, an adapter molecule can be used to generate sticky ends. Alternatively, an endonuclease can be used to generate sticky ends. So this is some other another approaches when we are doing the mechanical shearing of the DNA to make the fragments. Okay. Next, then the transformation is done. So transformation is the post ligation clones are transformed in a suitable host to get the colonies. Again, as I told you, depending upon your requirements, that what are you supposed to do after cloning for the like protein purification, or you want to check some kind of an interaction or something. So for that you choose accordingly what kind of a cells you want, what kind of a vector you want. So accordingly, post ligation clones are transformed into a suitable host to get the colonies. A suitable host can be bacterial strain or yeast. Different methods of delivering clone into the host cell is discussed in the future lectures, and that we will see in the future lectures. Okay. So this is the table that is showing the carrying capacity of different vectors, that how much MB of how much MB is the carrying capacity of different vectors. Okay. Uh, Next is the construction of cDNA library. So how it is done? Basically, it is done by first of all you get because it is a cDNA, that means a complementary DNA. So it must be made from an mRNA. So this mRNA is isolated from the cell, the whole mRNA content of that cell. That is then further used for transcription. Reverse transcriptase enzyme, as we have discussed before in the previous lectures, that it is an enzyme that converts, that forms DNA complementary to the mRNA, complementary to the RNA. So this the process is called reverse transcription, and the enzyme used is called as the reverse transcriptase. Okay, so it will lead to the formation of the DNA, which then when we get the DNA, we'll insert it into the bacterial plasmids, and after getting it into the bacterial plasmids, we'll transform that in the bacterial cells, and then after transforming them in bacterial cells, we'll grow them, isolate them, uh, and then plasmid purify them, and that is how we get our cDNA library. Okay, so when we are using mRNA, then there should be a method, because in mRNA the common thing here is the poly A tail. Okay, so we should have something common when we are using mRNA, so that all of the mRNA can be reverse transcribed. So in that case, that is uh, the poly A tail here, in the homopolymer tailing method. So we'll design the primers complementary to this poly A tail, that is the oligo DT primers, and these oligo DT primers will then be further used for the amplification, for the formation of the cDNA from the mRNA. Okay, next is the Gabor-Hoffman method. In this method, after first strand synthesis using oligo-DT primers in the presence of reverse transcriptase and DNTPs, DNA-RNA hybrid is treated with RNase H to produce nicks at multiple sites. Then DNA polymerase is used to perform DNA synthesis using multiple fragments of RNA as primers. So here we have used the RNA after formation of the single strand of this DNA from this oligo DT primers. Then these RNAs will be further treated with RNase H to make the nicks, and then those small RNAs will be further used as a primer for the formation of the further DNA, for the amplification of the DNA and making the cDNAs. Okay, then after the screening is done, so screening can be done by DNA sequence by the expression of the particular protein with immunogenic epitopes, or it can be done by the enzymatic activity also. These are three methods for screening. Then the when we are saying the screening by DNA hybridization, so generally a replica plate is made from the original plate, the original plate in on which we have gotten the bacterial colonies. A replica plate is made so that we have the original stock also. We should know what is the location of that original stock, so that whichever colony we get positive of, we can take the original stock from the original plate and can further use it for future use. Okay, so an original genomic or cDNA library is precious and will be consumed in later stages. So all procedure is performed with the replica plate. Original plate is kept safe as it was. The replica plate is further used for the purpose of the screening or whatever we want to do. Okay. Then the clone is transferred on the nitrocellulose membrane with retaining identical pattern of colonies on the master plate. The cells on the membrane are then lysed and released DNA is denatured, deproteinated and allowed to bind the membrane. Then the labeled probe prepared in step 1, that will be added in step 1. We prepared the probe. I didn't show it here. So probe will bind to the target DNA due to base pairing, and the membrane is washed to remove unbound probe. Okay, so here the unbound probe will be removed. The bound probe will tell us the sequence of the DNA to which it has bound. Okay, so the position of the label probe is detected by autoradiogram, and the position of signal on membrane can be matched with the master plate to get the location of the corresponding colonies. ठीक है, अब अगर हमें इम्यूनोलॉजिकल मेथड से स्क्रीनिंग करनी होगी, so in that case again we have to make a master plate. Then the cells will be lysed and bind protein to the matrix. Okay, cells अगर lyse कर देंगे तो जो protein content है that will bind to the matrix on which the cells were grown, on which the cells were transferred. Then after, the primary antibody will be used and the secondary antibody will be used. Secondary antibody will have something for the detection, and then the protein will be detected. So this is how the immunological method works, because here the antibodies are involved. Okay. In enzymatic method, the method is based on the ability of protein to exhibit an enzymatic activity. This method is not very specific but allow us to identify a class of protein with non-enzyme. So basically this method is generally useful when the particular protein has some kind of the enzymatic functions. Okay. Then the isolation of genes. Once the position of the clone is known, it is extracted from the master plate and plasmid is isolated. In few cases, clone is further diluted to check the homogeneity of the clone. The purify of the clone and presence of clone is further tested with a PCR. So PCR here comes handy. Just a minute. एक minute. So basically, PCR here is here used to further amplify or test the positive clone. Okay, now coming to, we have amplified the gene, we have gotten a basic idea how cloning is done. Now we have gotten our gene also. Now, what about the vectors on which the gene must be cloned? So, vector DNA has two main responsibility: ability to carry foreign DNA and ability to replicate in the host cell. That means it has its own origin of replication and other things required for the replication inside a machinery. Okay, raw material it can get from the cell, but it should have that machinery. Hence, to fulfill these responsibility, a number of properties are desirable. So the criteria of good vector is it should have a low molecular weight so that the uptake is easier. Post entry into the host should give uh, phenotypic changes so that we can come to know that the vector has entered the cell. Okay, multiple cloning site with unique restriction site so that we can use suitable enzyme to clone our gene. Okay, and high copy number. High copy number होगा तो we can over express a particular gene inside the cell. Okay. uh, these were some of the desirable properties, the criteria for a good vector. Now the components of the vector, what should be there in the vector? So it should have an origin of replication so that the replication can start. Uh, it can have that point from where the replication must start. Then the selection marker can be some kind of an antibiotic resistance or some kind of an enzymatic gene which is essential to give the phenotypic change in the host after the entry of that particular plasmid. Okay, and it should also have a promoter so that the gene replication can happen smoothly. Okay, so the different type of vectors that can be used for cloning are: as vector needs to replicate in different host strains, vector needs special additional structure features to make it suitable for a particular host strain. Why one vector doesn't replicate in different host strains? Because replication of vector DNA is controlled by origin of replication, and it needs to be recognized by the host factor, especially DNA polymerase, to perform the replication. That is why it is very very important that what vector you are using and what kind of a cells you are using. ठीक है, so there are different type of vectors, the DNA vectors in which the cloning can be done. It can be bacterial plasmid, it can be a phage based vectors, it can be yeast vectors, or it can be mammalian vectors. ठीक है, so bacterial plasmids are widely been used for cloning foreign DNA into the bacterial or host as host strain. Different forms of plasmid can be, sorry, bacterial plasmid is a double-stranded circular DNA, exists in three different forms. If both the strands of circular double strands are intact, then it is called as the covalently closed circles. Whereas if one of the strand has nicked, then it acquires a conformation of an open circle DNA. And during the isolation of plasmid DNA from the bacteria, covalently closed circular DNA loses few number of turns, and as a result it acquires the supercoiled configuration. So these are the three forms in which a bacterial plasmid generally exists. Okay. If we'll talk about the eukaryotic vectors, then we have either yeast vector, the phage based vector, or mammalian vectors. Okay, so vectors are generally an extra chromosomal DNA. They are not the part of the main chromosomes, and there are some kind of integration vectors are also. ठीक है, so eukaryotic plasmids, if we'll talk about the yeast vector system, they all have couple of similar features such as presence of MCS, that is multiple cloning site, shuttle vector, origin of replication of E. coli and yeast, and presence of selectable markers, so that we can know that what gene has gotten our plasmid and what not. Okay, so there are three types of yeast vector: the episomal vectors, the integration vectors, and the yeast artificial chromosome. We are again not going into the details of these vectors. I hope you guys have read it from the lecture. It was just to give you an overview so that you can just recall what we have read. Okay. Eukaryotic plasmid mein another thing that we have is the baculovirus. Just another thing that we have is the baculovirus vector. So baculovirus is a root-shaped virus infecting invertebrates, including insect cells. Okay, so that is baculovirus. That vector can also be used in case of the eukaryotic cells. Eukaryotic vectors, another category is the mammalian vectors. Okay, that can also be used for cloning. Uh, then the bacteriophage vectors can also be used. That is, bacteriophage vectors are generally used in case of the phage display when we are doing the phage display technique. But yes, bacteriophage vectors can also be used for cloning. Then after what now we have gotten the information about our gene, our vector, we have cloned them also. Now how they should be transformed in the suitable cell so that we can get many copies of that, and when we can also keep them for the future use also, for overexpression and whatever we want to do. So for that, the process of transformation comes in handy. So in transformation, what you do is you will transform your cell, your vector inside the suitable cell. So transformation is a process by which cell-free DNA is taken up by another bacteria. Then the DNA from the donor bacteria binds to the competent recipient cells, and the DNA enters into the cells. Okay, and it is not it's not like it will take up your DNA as such only. It must be prepared in a way. The cells must be prepared in a way that they are ready to take up your DNA, and also keep that inside the cell so that we can use them further. Okay, so the DNA enters into the recipient cell through an uncharacterized mechanism. The DNA is integrated into the chromosomal DNA through a homologous recombination, sometimes, not always, and naturally transformation is common between closely related species only. Okay, so if we'll go by the natural process, then it is generally happens between the closely related species only. Okay. So the list of selected agents has potential to make the competent cells. So these are the different cells, and these are the different agents which help those cells to be competent enough to take up the DNA, the desired DNA. Okay. So preparation of competent cell, generally bacteria is incubated with a divalent cation, calcium chloride, magnesium chloride, or rubidium chloride for 30 minutes at 4 degrees Celsius, and during this process cell wall of the treated bacteria swells and gather factors required for intake of DNA docked on the plasma membrane. ठीक है, so the treatment is very very important with this divalent cation. ठीक है, and generally the cells of the log phase or exponential growth phase are the ones that are desirable for the making of com cells, so that whenever you'll further grow them for your purpose, they can easily grow and they won't die while taking up the DNA and growing further. Another method for the transformation is the electroporation technique. In this, the plasma membrane is composed of the lipid and protein, we all know that. And these macromolecules give a partial conductance to the cell membrane. When a high electric pulse is given to the cell, the charge run across the membrane and partially disturb the arrangement of the lipid molecule. As a result, it makes formation of the pore and allow easy passage of the macromolecule, specially charged molecule like DNA, into the cell. And after the electroporation, cell is allowed to recover from the damage and it forms the colony on the selective solid media. Okay, so electroporation is generally charged based technique. When we are transforming in yeast, then we have another different method, that is the lithium acetate method. So it can be used in case of the yeast. We are again not going into the details of the different methods for transformation into the yeast or something. I hope you guys have read it from the lectures. Then we are going for the blue white screening. Now we have transformed the cell with the desired vector. Now we want to screen the cells. Okay, just a minute. Hmm. Now we want to screen the cell, that which cell has gotten our plasmid and which does not. So for that many methods are there. One is the blue white screening. Okay, so it is generally a chromogenic substrate. So the use of chromogenic substrate to detect a particular enzymatic activity is the basis to screen the desired clone. So the most popular system is the blue white screening, where a colorless substrate is processed to a colored compound, and the colorless compound is X-gal or 5 bromo 4 chloro 3 indole beta D galactoside. Used in this screening method is a substrate for beta galactosidase. Beta galactosidase is the enzyme. ठीक है. So the enzyme beta galactosidase is the product of lac-Z gene of the lacoperon. It is a tetrameric protein, and an initial N-terminal region of the protein is important for the activity of the protein. So in this system, what happens is the host contains that lac-Z gene without the initial region, where a vector contains the alpha peptide to complement the defect. So basically, your host does have the lac-Z gene, but it is not the complete gene. To complement this defected lac-Z gene will provide an alpha peptide from outside in our desirable vector, so that it can complement that gene and it can form the enzyme beta galactosidase. Then this beta galactosidase will degrade this colorless compound X-gal, and it will give us the blue color. ठीक है. That blue color will define that our gene has transformed inside the E. coli, and now it can be used further. So it is just to screen the cloned ones from the uncloned ones. Okay, so this is how it is done. And another thing that can be used is the antibiotic sensitivity. So vector carries a functional selection marker such as antibiotic resistance gene to be used to select the clone. So the antibiotic resistance gene product has multiple mechanisms to provide resistance into the host cell. Okay, and in this approach, a circular plasmid containing antibiotic resistance can be able to replicate into the host cell plated on an antibiotic containing media. Because when bacteria is getting grown on the plate containing the media and the antibiotic, so it is taking up all the nutrients from the plate only. So obviously it is taking the antibiotic also. And if the bacterial cell does not have a gene which can resist this antibiotic resistance, then obviously it won't be able to grow in the presence of that antibiotic. So this antibiotic sensitivity is also one kind of a marker to tell us that yes, the transformation has happened, and it can be further used for whatever purpose we want. So this is how it is done. In case of the ampicillin, sorry guys, there is some issue. Okay, okay, okay. This is for chloramphenicol. Yes, so basically, ampicillin will make a gene product beta lactamase, which will help in the degradation of the ampicillin, and so that the resistance develops and cell is able to grow. Just ठीक है. So that is how cell is able to grow. If it is kanamycin resistance inside the gene, then the neomycin phosphotransferase 2 will form, that will lead to the covalent modification of the kanamycin, and now it won't be harmful for the cell. ठीक है. for the cell who have kanamycin resistance plasmid inside them, not for all of the cells. So that is how it will help in selection. We will talk about the tetracycline. So the ribosomal protection protein will be formed. So efflux of tetracycline outside of the bacteria will happen so that the bacteria can grow peacefully. Tetracycline won't affect the bacteria who have the resistance gene in them. Okay. Then the chloramphenicol acetyl transferase is the product of the gene, and this chloramphenicol will convert this gene will transferase will convert chloramphenicol to acetyl chloramphenicol so that the cells can grow without any hindrance. Okay, so this was about the antibiotic sensitivity. Okay. Next is the insertional activation. So in this approach, a foreign DNA is cloned within the coding gene responsible for a phenotype. As a result of insertion, the gene product is not available to modulate the phenotype of the host. This approach is known as insertional inactivation, and it can be used with a suitable genetic system. So here we are actually silencing a phenotype so that we can know if the phenotype is there, that means the clone doesn't have happened, and if the phenotype is not there, that means the cloning has happened. Okay. Next is the complementation of the mutation. So in this approach, a mutant host strain can be used to screen the plasmid containing the missing gene, and the transformants will grow only if the gene product from the clone will complement the function. In general, genes taking part in metabolic pathway or biosynthetic pathway are routinely being used for this purpose. Sorry. Okay, so there are three important requirements for this approach. One is the host strain should be deficient of that particular gene, so that if gene belongs to biosynthetic pathway, the mutant host in that case are called as the auxotropes and are as host dependent on the gene product or the final product of the biosynthetic pathway as a supplement in media for the growth. ठीक है, तो this generally is done for in case of the yeast, in which the uracil component is basically used to for this kind of a complementation, where the plates are generally made with or without uracil, so that we can know the effect of that particular gene. So that is how the complementation of mutation happens in case of the yeast system. Okay. So positive selection, negative selection. In case of the positive selection, host strain does not grow in the media lacking the functional gene, but the host transformed with the recombinant clone can be able to supply the gene product required to grow in the media. As I've told you, in case of yeast, uracil system is generally used. And the negative selection is, in the negative selection, a chemical compound is added to the media which will be converted to cytotoxic agent in the presence of a gene product, and as a result it does not allow the growth of the wild type cells, but host strain transformed with the recombinant clone has non-functional gene product. So basically insertional activation of that particular gene has happened and grown in the presence of the compound in the media, and when the insertional inactivation has happened, then obviously it won't form the cytotoxic product, and the cells will be able to grow. Okay, so this kind of selections are generally done when we are talking about the complementation of the mutation. ठीक है, the screening of transfected mammalian cells. So generally it is done by the reporter gene assay. In the reporter gene assay system, a chimeric construct is produced with an enzyme gene is cloned in front of the promoter of gene of interest. And the general reporter gene construct contains a eukaryotic promoter and an enzyme for easy readout. The reporter gene construct is transfected to the mammalian cell with a suitable transfection reagent as discussed in the previous lecture, and afterwards the cells are stimulated with the agent to stimulate the production to transcription factor to bind promoter and drive the expression of the reporter gene. A suitable substrate is added to measure the activity of the reporter enzyme. So basically this whole assay is designed, and a reporter gene here will make the enzyme. And if the transformation has happened, if the transfection has happened, then only the enzyme will be formed and the substrate will give an output product which will be further recognized. Otherwise the enzyme will not be formed, and the reporter assay will tell us that the transfection hasn't happened. Okay. Now we have seen what kind of methods are involved when we want to see if the transformation has happened or not, and if we have used the suitable vector or not, suitable cells or not. Now when we are selecting the vectors, there are two further categories of the vector. Either it is a cloning vector or it is an expression vector. So expression vector is generally used when we want to form the protein from the cloned vectors, and if we want to use them for other purposes like the replication, genomic or cDNA library formation, and further cloning related things only, then we'll go for the cloning vector. But if we want the expression of that particular gene, that we will go for the expression vector. Okay. So depending upon that what are our requirements, we choose the vector from that category only. If we choose the expression vector, then obviously we need to synthesize the protein also. We need to make the protein also from that. So generally what is the protein synthesis machine? So protein production is a multi-step process, and an overview of these steps are as follows. So the binding of RNA polymerase to the promoter element to start the transcription to form the mRNA. If mRNA will be formed, then only the protein will be formed. If no mRNA is there, then no protein will be formed. Okay. So as soon as mRNA is synthesized, the translation machinery start the synthesis of the protein. Protein synthesis start usually at the start codon AUG and ends at the stop codon, that is either UAA, UGA or UAG. These three are stop codons. Any of them can be there for as an indication of the stop codon. Okay. In bacterial, transcription and translation occurs simultaneously, and translation starts as soon as the mRNA comes out from the RNA polymerase. So in bacteria generally transcription and translation are happening together, as we have read in the previous lecture. So that will happen here also. As soon as the mRNA comes out from the RNA polymerase, the translation process starts. Okay. Then the depending upon the host and the vector, the cells are used, as I've told you. Now here, when we are going for the protein synthesis machinery, when we are actually want to express a particular protein, then the host production machinery, the cells which should be used for an expression system, should be used properly. If we want the protein from the E. coli, from the yeast, from the insect cell line, or from the mammalian cells, depending upon our requirement, if we want post translation modifications or not, and if we want different kind of the features that get added in the eukaryotic system or not. Okay. So depending upon that, the host production machinery used, E. coli, yeast, insect cell and mammalian cells, any of these cell lines can be used for the expression system. Okay. Now the criteria to select an expression system is the quantity of the desired protein. What is the quantity of the desired protein that we need? Size of the protein. What is the size of the protein? Generally E. coli expression system is not preferred for the large size of the proteins. Okay. In that case eukaryotic expression system is more suitable. Then the compatibility between the source organism and the expression system from wherever you've gotten the gene. Then the downstream application, because this is one of the most important criteria, what you want to do with your protein. Okay. The protein production can be for generation of the antibodies, or any expression system may suit well for this purpose. But if the protein is required for the activity or for the ELISA, then a compatible expression system is preferred. Otherwise you won't be able to see the activity. Okay. So for that there are many criteria depending upon which you should select your expression system. Okay. So the typical components of an E. coli expression system are uh, the promoter region, an upstream sequence to the gene that provides the docking site for RNA polymerase, ribosome binding site, a termination site, and an affinity tag. It can be either a his tag or a GST tag, S tag. There are many tags that can be used in case of the protein that is already there present in your uh, expression vector. ठीक है, that gets attached to your protein, your desirable gene, and whenever you want to purify, you can purify it using affinity chromatography. Okay. Then there is an IPTG inducible promoter. If you remember in the previous lectures, we have learned about the many kind of the operons, the tryptophan operon, the lac operon, and all. So IPTG can be help in the uh, it is actually a synthetic analog of the lactose, and that is why it can be an inducer for the lac operon, so that uh, it can be used for the overexpression of your desirable protein. So the lac promoter is very widely being used to construct different expression plasmid to express the protein in E. coli. The different vector contains lac promoter or its derivative. So the lac promoter, uh, it should have a lac promoter, the tryptophan tag promoter, or the tryptophan TRC promoter. So it is an high tag promoter is basically a hybrid promoter of tryptophan and lac, and uh, जो TRC है that is also similar to the tag promoter. So basically this IPTG inducible, these are the three promoters which can be induced using the IPTG and which can be used in your protein expression system for the overexpression of the particular protein. ठीक है. Then we have the bacteriophage T7 promoter also. Again, we are not going into the details of what kind of a promoter what are. Okay. Then the factors that affect the protein synthesis in E. coli. So basically the translation efficiency is governed by composition of the promoter, especially the sequence of Shine Dalgarno sequence, which enables the binding of the ribosome protein production machinery. ठीक है. Then so basically the translation efficiency, the growth condition, these are some of the factors that affect the protein synthesis in the E. coli. Next is the expression of the fusion protein in the E. coli. So advantage of fusion protein is easy purification, targeting to the cellular compartments, half life of the protein is good, and the solubility is also keeping a tag at n terminates direct the protein synthesis and helps in increasing the solubility of the foreign protein. So generally it is desirable that you have a protein tag at n terminal so that it increases the solubility of the protein. ठीक है. So there are some, this was the thing that we have learned in the last week. Different kind of expression system, different kind of vectors, how the cloning is done, what are the factors required for cloning and all. Now we'll come to the assignment questions. So this will go in a way that has been going always. I'll ask you a question and you guys will write the answers in the chat box. No one will speak up the answers. Okay. So we'll start with the question 1. Dash is an example of restriction digestion enzyme. Just a moment, class. Huh? Okay. So yes, the answer is C. That is EcoR1. Correct. It is a restriction digestion enzyme. ठीक है. Next is, other than restriction digestion enzyme, genomic DNA can be fragmented using what? If you don't want to use restriction digestion enzyme, then what is the other thing that can be used to fragment a DNA? Yes, everyone answer. बाकी लोग कोई आंसर नहीं करेगा? Yes, please. Yes, it is mechanical shearing through which we can do so. Okay. Next is, dash can be used to generate sticky ends during the cloning into a suitable vector. स्टिकी एंड्स कैसे बनेंगे? हाँ जी, स्टिकी एंड्स कैसे बनेंगे एवरीवन? आंसर यस। बाकी लोग? Exactly. It is an endonuclease that can help in the formation of the sticky ends. Right. Okay. Next is question number 4. The first step of construction of cDNA library is. cDNA library we are talking about. So the answer should be. Everyone. Yes, it is the isolation of mRNA. Obviously, when we'll isolate the mRNA, then only we'll able to produce the cDNA. Okay. Yes, the answer is isolation of mRNA. Next question. Homopolymer tailing exploits the presence of dash present on mRNA to synthesize the first DNA strand. What is that? Question number 5. Answer is. बताइए होमोपोलिमर टेलिंग में क्या होता है ऐसा on mRNA which helps in the synthesis of the first DNA strand? Answer is yes, it is poly A tail. तभी तो हम oligo DT primer use कर सकते हैं. So it is the poly A tail. Okay. Next question. Dash can be used as a selection marker in molecular vectors. Arpi Das, you are answering for question number 5 or question number 6? Okay, okay, perfect. बाकी लोग? No one wants to answer. मैं बस एक सेकंड में आई. Yes, the answer to this question is. Yes, the answer to this question is the presence of antibiotic resistance gene. Okay. So, yes. Next question. If one of the strands of bacterial plasmid has a nick, then it requires the confirmation of what? अगर इसमें एक nick होता है, then question number 7, everyone. Kumar, answer for question number 7. No one wants to answer. Yes. Yes, so the answer is open circle DNA. ठीक है, that will be called as open circle DNA. Next question. During transformation of bacterial cells, the free DNA is integrated into the chromosomal DNA through a what? Integration into the chromosomal DNA we are talking about. So the answer to this question is yes, it is homologous recombination. Right. Next question is, it is preferred to use bacteria of dash for making competent cells for transformation. What phase? बताइए. Question number 9 I'm talking about, guys. Yes, it is the log phase that we need to use the bacteria of. ठीक है? Next question. Question number 10. Uh, Dash is one of the DNA delivery methods in mammalian cells. What is that? Yes, it is electroporation. Next question. Beta lactamase gives antibiotic resistance to bacteria against what? Beta lactamase. It is ampicillin. We have seen the table, right? Next is the approach where cloning of foreign DNA is modulated to affect the phenotype of host is known as phenotype of the host. Question number 12 I'm talking about, guys. Yes, it is the genetic engineering. Okay, next question. During the Sanger method of DNA sequencing, chain elongation steps chain elongation stops at DD and TP due to what on the DD and TP? 13th. Yes, it is the absence of 3 hydroxyl group. That is why it is called as the dideoxy, because एक deoxy तो उसके पास था ही, 2 hydroxyl group तो उसके पास था ही नहीं, अब 3 hydroxyl group भी नहीं है. That is why DD NTPs. Okay, question number 14. IPTG is a synthetic analog of what? Yes, it is of lactose. ठीक है. Next question. One of the advantage of fusion protein expression in E. coli is yes, it is the easy purification. So with this, we come to an end of our question answers of assignment. If anyone wants to ask me anything, they can. Guys, just a moment. In question number 12, I think we went for a wrong answer. The answer should be insertional inactivation we did for genetic engineering. So the correct answer is insertional inactivation, because we are affecting the phenotype of the host. So the answer is B, not the C. Okay. I think I spoke it wrong, and you guys also went for wrong answer. So the answer is B, insertional inactivation. Okay, so anyone has any question, they can ask me. Any question, anyone? I hope you guys understood the lecture and you guys understood the videos also. So we have two more to go. Actually, three more to go. Three more lectures, and then the doubt sessions will be over for the subject. If you have anything, you can ask me. You can ask me in the next lecture also. There's no issue on that. And if you don't have any questions, you guys can leave the meeting. Thank you for joining. You're welcome, Arupi.